All right, thank you for coming, and I'm happy to be here today, and I'm going to tell you about this model called Capricorn that we've developed. This was work by Tongqi Fang, who is an incoming PhD student in the computer science department, working with Sheng Wang, who's an assistant professor there, and with me. Unfortunately, Tongqi was not able to get a visa to get here today, so I have to give his talk for him. But he actually did a lot of the work. There are some other authors, of course, that are acknowledged at the end. And he also developed these slides. So a lot of this background I can go through, whizzing through, because Jian did a great introduction before. You all know that the DNA adopts a complex structure, and you also know that there's this Hi-C assay that measures this 3D chromatin structure. The important point is that the output of Hi-C is a matrix in which rows and columns correspond to DNA loci, and the values represent counts, which is how frequently those two loci are proximal to one another in a population of cells. So we're talking about bulk Hi-C in this work, as opposed to the single cell from the previous talk. One thing in particular that we're interested in are different features of the chromatin. And again, Jian mentioned some of these, like compartments and domains, and particularly fine scale elements called loops. Just to be clear, a loop, as illustrated here, is sort of like a small Gaussian hump in your 2D matrix. So the circled region there corresponds to a loop, and it corresponds essentially to two distal regions of the DNA that are coming into close contact frequently in many cells in the population. And these are interesting, not just loops, but also other aspects of chromatin 3D structure, because they have important implications for various processes in the cell, disease processes, gene regulation, and so on. So in this talk, we particularly are interested in chromatin loops, partly because they do have there's sort of more direct implication in some of the gene regulatory mechanisms. And so it's important to be able to detect these loops accurately. One of the challenges is that seeing a loop in a Hi-C matrix typically requires a lot of sequencing. It should be obvious that if you're sequencing if you're looking at all pairs of contacts in the genome, then the sort of coverage scales quadratically, right? In order to get twice as many counts per locus pair, you need 4 times as many reads, right? So Hi-C can get expensive. And in fact, people didn't really describe loops until, well, I typically think of it as the 2014 paper by Rao et al. From Erez Lieberman-Eden's lab. So the original paper came out in 2009, although I noticed Jian had a loop citation from 2013, so maybe they were talking about it a little before then. And the point is, if you look at one of this, like this Hi-C matrix, I don't know if you can see it, but there's a little circle over here that corresponds to a loop, but you can see it's kind of hard to see. And then if you take that same data and you sequence it more deeply, then the loops start to become apparent, which is partly why they didn't really get described in the literature for 5 years, because the original Hi-C matrices were all low depth matrices. Okay, so one solution to this problem is to pay a lot of money to your sequencing core and sequence all your Hi-C libraries to billions of reads. Another solution is to train a machine learning model to hopefully save some of your research funds to do other things. And the idea is that you could take a low coverage matrix and rather than pay to sequence it more, you give it to your deep learning model and it learns to enhance it. And to train these kinds of things, you take a high coverage matrix, you downsample it to get a low coverage one, and you get many such pairs, and you train models to interpolate between the two. So this is a well-studied problem. There's generally 2 categories of methods that have been developed to address this task. On the left are methods that use convolutional neural networks. There's like 5 of those, and then 6 of them that use these generative adversarial networks. If you don't know what those are, it's okay. I'm not going to talk about them in any detail, but suffice it to say 2 different classes of deep neural network models. What we're interested in today is thinking about particularly how we can inform this kind of deep learning architecture to try to take into account more of our prior knowledge about what are the biological aspects of the data that are important. So many of these systems are trained using something like mean squared error, where you're just trying to say, I've got my ground truth, which is the actual Hi-C matrix and high coverage, and then I've got my predicted enhanced matrix from my model, and you take the mean squared error between them. But in this particular example, if you have 2 different predictions, the mean squared error is going to prefer the top one. On the other hand, if we take a loop caller and we run it on the actual observed data and on your prediction. In this particular case, the second one, the loop caller, makes much better predictions. So if you estimate how good this model is in terms of sort of the proportion of recovered loops, then you prefer the second one. So we want to have a model that will do both of these things simultaneously. So the main contributions of the Capricorn model are, on the one hand, we're going to do an optimization that uses multiple views of the high-C data to try to draw out different aspects that we think are important. And then we're also going to use a different category of machine learning model called a diffusion probability model. So just as a recap, remember the existing methods train using these low coverage to high coverage pairs, where you downsample a high coverage to get a low coverage pair. In the Capricorn setting, instead of just having a single input matrix, we sort of expand that matrix to give different views. So in this picture, we have 5 different views. You have the original view of the matrix. So we do actually use the MSE as one of the terms in our loss function, because we do care that we actually have a low mean squared error. But in addition, we do things like this. So for example, we know that the random polymer behavior of DNA gives you a strong diagonal in the high-C matrix, and sometimes the mean squared error gets carried away and just says, just make sure you match that diagonal trend, which isn't super interesting. So you make a distance-corrected version of the matrix. The next one captures domain structure, so these topologically associated domains. You can call domains using an algorithm called the insulation score algorithm. And an intermediate step in that is this matrix that basically each entry is now not a count, but a score of sort of how domain-like we are at that particular position. And then there are 2 different scores that are derived from one of the first loop calling algorithms. The algorithm is called hiccups, and it has a score that is sort of the relative ratio of the current pixel to the local background with respect to this model that hiccups uses. And so it's sort of how loopy are we at this particular position. And then there's a p-value version of that where there's a null distribution to tell you how significant is that loopiness. Okay, so we're going to take all of those as input and also all of those as output. The other piece that we're going to bring to the table is this idea of using a conditional diffusion model. So this is not something that we invented. We're just using this technology. The idea is illustrated here, interpolating between a high resolution image. In this case, it looks like some kind of junco maybe, or no, it's a chickadee. A high resolution image of a chickadee and a low resolution image of a chickadee, and then interpolating between them. And the way a diffusion probability model works is you basically have 2 different diffusion modes, and it's a supervised model, so it's being trained to go from one to the other. It's been shown in many different situations that these diffusion models work really well. So they achieve state-of-the-art results in image generation, in painting, and of course in super resolution for other types of images. So in Capricorn, we take all of these views and we do a diffusion process. The diffusion actually operates on the main matrix, but then you make all the views from that main matrix on the output as well, and the loss function incorporates terms for each of the different views. Okay, so then we had to test Capricorn. There is always some uncertainty about the best way to do these kinds of tests, and if you look in the literature, people do it different ways, so we just try to do it all the ways, so we made sure we cover all the bases. One simple way is what's called a cross-chromosomal setting. So you take the genome, divide the chromosomes into training chromosomes and test chromosomes, train on some, test on others. And that's pretty straightforward. It's a little bit unrealistic because you don't typically do a high-C experiment on some chromosomes and then look at the other ones later. And so another setting that you can do is the cross-cell type setting, where you can basically say, I'm going to train on one cell type, and then I'm going to predict in a completely distinct cell type. That also makes sense. The problem is sometimes you can learn features of the actual chromosomes, and those will generalize between. And so to solve that, you can do a combination of the 2, where you basically say, I'm going to do a cross-chromosome and cross-cell line setting, hold out some chromosomes from the training cell line, and then use those only in the test cell line. And then in terms of the score function, we have several different ways to evaluate the model, but one of them is to ask, how well do we recover the loops that we would have gotten if we'd had the access to the the original high-resolution data. And so we do that by running a loop caller and then measuring the F1 score, which many of you are maybe familiar with, but just as a recap, you can typically, if you have a predictor, you can define the precision, which is sort of how many of the predicted loops are true, and your recall, which is how many of the true loops are predicted. And if you take the harmonic mean of those two, then you get this score called an F1. And so we take the loop caller on the real data, the observed high-resolution data, and the enhanced, and then compute the F1 score between them. So with that background, starting with the simplest setting, the cross-chromosome setting, we can see that we get significant improvements in loop detection. So in this one, we've compared to state-of-the-art methods. So there's two different variants of the high-carn method, whether it uses a CNN or a GAN. There's also high CSR and high CNN. And we also included as a control the low coverage. So if you just run this directly without, just on the low coverage data. And so what you can see is in all of these situations, you get a significant improvement from Capricorn's approach. You also get the same kind of thing if you do if you split the, if you run this test in a different cell type. I didn't show the plot here. And then we also looked at this in the cross cell type prediction setting. So now we're training in K562 cell line and then testing in the GM12878. And again, you see a consistent trend here where we're significantly improving. And here we actually did this in the previous one. We were doing all of the loop calls using one loop calling algorithm. Here we tried it with 3 different loop calling algorithms: hiccups, chromocyte, and mustache. And the results are consistent across all those different algorithms. And then finally, looking at the cross-chromosome and cross-cell type setting, you see similar kinds of trends, although this is a harder setting. And you will see, like, for instance, in the right-hand plot, there's some cases where just doing the enhancement seems like a bad idea for everyone, that you actually get better performance from running the loop caller on the low coverage data. So to emphasize that it is a hard problem. One of the things we wanted to be sure of is that we didn't just do well on things like loop calling. So we went back and measured the mean squared error. So this is just showing that by incorporating a loss function that has mean squared error plus all these other terms, we don't necessarily discard our ability to do well by mean squared error. So this is just showing that we're improving in that realm as well. And then finally, to illustrate that this idea of using multiple views of the data is generalizable, we went back and incorporated this into several of the other models. So we took high CNN, high CARN 1 and 2, and high CSR, and we modified them in 2 ways. First, to just take all of these different views as input, and then also to take them as input and output. And what you can see is that with the exception of high CSR, and honestly, I'm not really sure why it didn't work in that situation, but for the other ones, adding these different views into these other models also does a good job of boosting their performance. So it suggests that the idea generalizes beyond our particular model. Okay, so that's the Capricorn story. Overall, adding 5 channels to the resolution enhancement and then using this diffusion backbone, both of those seem to be important and generalizable. And in the future, we're looking at incorporating other biological views at different scales, thinking about incorporating temporal information, like for instance, for time series from cell development, and then also looking at single cell data as well. And as I mentioned at the beginning, this work was primarily by Tongqi Fang, but also with important contribution from Yifeng, Adi, and Tongqi's advisor, Sheng Wang, and of course, all the other co-authors as well. So thank you all for your attention, and I'm happy to take questions.
With this 5-view, so does the loss function, can it be attributed to each of the views separately, and does that, what are the relative proportions? Do they change? Is it that some of the views are easier to train than others?
Yes, it's a great question. Yes, the loss function, it's just a sum of terms with each of the views represented by one term. And there is a dynamic process to basically adjusting the relative weights to ensure that the contributions to the loss are approximately equal throughout the training. Because otherwise one of them can just dominate. Yeah. Yeah, I can hear. just seemed like you had to. Yeah. Oh, I see. I thought you meant in terms of network layers. And how shallow it's, yeah, good question. So I think for this, we've been doing what most people in the literature were doing, which is a 1 to 16, a 16-fold ratio between the input and the output. But what we're doing now is working on things where you can actually, we're going to train on many different ratios jointly and have the desired output ratio be part of the input. So we would train a model that knows what it looks like to go by a factor of 2 or 4 or 8 or whatever. And then the user could say, give me a 2 or a 4 or an 8. I think that's an interesting direction for the future. But all this work was at a 16 ratio. Yeah, I think that mic is off. Quick, run, you got those. Yeah, they won't be able to hear you.
So let's take all the questions on the side. My question is, because we know the genome, if you add the sequences, additional features to the prediction, like from your perspective, how much you can.
Yeah, it's a great question. So what value will there be in adding the sequence? I mean, I think this is super important and something that we're actively working on. The intuition that if the model could learn about what kinds of structural elements lead to loops, for example, we know that in some of the higher resolution data that ARES has produced recently, you can localize these loops down to tens of base pairs, right? And so sequence elements should be super valuable for helping with this kind of enhancement. I don't have any empirical results to back that up yet, but that's what we're working on.
As the moderator, I give myself the last question. So the multi-view aspect of it seems like it is very easily generalizable to different data modalities in terms of like within maybe chromosome conformation capture or like GenDet, maybe even across imaging. And so are you doing anything along the lines of maybe highest, like different methods capture different things better, like micro-C has better detection of regulatory loops, high chip detects targeted loops. So in your framework, it seems like very naturally you can have multiple different data sets from the same cell type using the strengths of all of them, and then imaging you can throw in. So is there a limitation to how many views you can have inherently, or it's?
Oh, I see. I was going a different direction there. You could train jointly from all these different kinds of input data types, but you're asking about, is there a limitation on?
No, like that last question separate, I guess.
Oh, okay.
But, I mean, last question is because if you put a lot of different data types, maybe it's going to be a lot of views, right?
I mean, I tend to think that a view is sort of an intuitive way for a human to visualize what all we're doing is designing a better loss function that incorporates our prior knowledge, right? And so in the end, what the model sees is it's a different loss function. It happens to be that we can visualize it in these nice 2D matrices. But I think you're right that if you had other kinds of 3D chromatin data, you could imagine saying, okay, these we need to tailor even other views for. And then I hadn't thought about it, but you're also right that you could imagine having a model jointly that can enhance, because now there's 6 or 7 or 8 different kinds of these Hi-C-like matrices. And in principle, with enough data, you should be able to do all of them in one model, which would be cool.
Thanks, Phil. Thanks a lot.